Jamie Thompson was here, John Gearhart, a lot of people. But what was nice was that Ian Milton was here for the very first meeting, so it's great to welcome him back again for this 6th stem cell meeting on the Mesa. Ian is probably best known as the leader of the team that cloned Dolly the sheep. He's now at the Centre for Regenerative Medicine at the University of Edinburgh, and the rest you can read about in the programme. So without further ado, Ian Milton.
Thank you for that very warm welcome. It's a great pleasure to be back here. I've really enjoyed today and congratulate the people who are concerned with the new venture next door and wish them every success. I thought what I'd do, particularly when I was shifting to this end of the day session, was adopt rather a philosophical approach and look at some of the background and history to attempts that people have made to change the fate of cells, to understand the mechanisms and the limitations of our ability to change cells, as well as summarise briefly what we can do now and take a little personal peek into the future by making some suggestions as to what we might do next. Sorry, I think I've twitched too enthusiastically. If anybody wants to get in touch with me afterwards, I'm sure I'm easy to find, but there's the email address, and that's the web page for the Centre of Regenerative Medicine that I work in these days. It bears some resemblance to the one we were hearing about earlier. It's a new building, we've just moved in. It's got lots of glass walls, it's got lots of core facilities, and I hope that some of you will visit in the future. So I'm concerned with two ways of changing the fate of cells: cloning, as you might expect, and the introduction of proteins, which might be expected to change the fate of cells. So let's think about cloning. Why did people think about cloning? What was the original reason for thinking about that sort of experimentation? It actually goes back as far as the 19th century, to the time when people began to ask the question, how is it that all of the cells that make up an adult body come from that single cell of an embryo? And one of the hypotheses was that as tissues formed muscle, let's say, they actually lost the DNA which was responsible for becoming nerves or any of the other tissues. And that, of course, leads to the prediction that if you take a specialized cell, that nucleus will have lost the ability to form an entire offspring, because some of the genes, some of the genetic information will have gone. Now, of course, this is an awful long time back, and the ability to do things with embryos was pretty limited. And this is a very early stage of development. On a number of these slides, I hope you've spotted already that I've often put the date in of the experiment, or the thought in some cases, and the name of the author of the experiment, the lead author. So this is an experiment with sea urchin at the four-cell stage, where what he did was simply to separate the 4 cells and ask, was each of those capable of becoming a normal offspring? And indeed it is. So what that means, it's a very short period, of course, but what that means is that those cells have all retained all of the genetic information. To nudge a little bit nearer to what we now think of nuclear transfer, a classic experiment by Hans Spemann with salamander embryos. And this is, I'm sorry it's grainy, but it is a copy of his original notebook diagrams. So what he did was to take the hair from one of his very young children and tie it around the embryo to put the nucleus into one half and therefore create an enucleated segment out of the other half. And he waited until the segment with a nucleus in it divided 2, 4, 8, 16 cells. And then he released the hair and allowed one of the nuclei to migrate across, closed the hair, and separated the two into 2 distinct embryos and asked, was he going to get 1 salamander, or 1 and a bit, or 2? And as you can make out, if you look carefully at the last diagram, there are 2 head to tail there in that egg. So even at this stage, there was no evidence of any loss of genetic information. They were all able to support normal development to term. This was in the 1920s. 10 years or so later, he described a fantastical experiment, as he called it, something which we would now recognise as being nuclear transfer. But at that time, because of the very small sizes we're talking about, the equipment simply wasn't available to carry out the experiment. It wasn't for another 20 years or so that people were first technically successful in cloning frogs. Now I imagine that you're all familiar with this basic idea, but just to make sure, the basis, the concept of nuclear transfer is that you have one egg. Oops, sorry. This is way too complicated for me. Certainly at the end of the day. You have one egg from which you remove these chromosomes. This is a mammalian egg that I would have studied, where the eggs are at the second metaphase of meiosis, so you're removing the spindle. So you remove whatever chromosomes are in that egg. You then, of course, need another cell which will provide the nucleus which will control development. In the case of Dolly, mammary tissue from her ewe is 2/3 of the way through pregnancy. And you can use the same pipette to put that cell into the perimetaline space, the space that you can see here between the outer shell, the zona pellucida, and the egg cell itself, the vitellus. This is really a cell fusion process, because what we would do would be to use an electric current to fuse these two together as a way of introducing the nucleus into the egg cytoplasm. It will go through, the nucleus will go through dramatic changes, it will swell very quickly, and on some occasions it will develop to become a normal offspring. A lot of the details are different with different species, but that's the fundamental concept for all of the experiments. The first species to be worked with successfully were amphibians, and this is a classic picture of an experiment of John Gurdon's, where you can see that he had eggs which were taken from this green animal, and nuclei which were taken from an albino animal, so that when he transferred the nucleus into eggs from this animal, the offspring that he got were all albino. The first attempts were made taking nuclei from early cleavage stages, and they were successful, so naturally they progressed and took nuclei from later stages of development. And what they noticed was that the efficiency dropped as they worked through progressively into differentiated tissue. And this is a fact which is not always made plain, that there have never ever been any clones of adult frogs. There have been tadpoles produced if you took a nucleus from an adult, and adults formed if you took cells from tadpoles. So I repeat the point, there have been no adult amphibians produced by nuclear transfer from another adult. Quite what the difference is, quite what it is, that observation's decades old by now, and I don't think people have any real idea at all as to what the difference is between those two stages, which is so critical for that last stage of development. So people began to think of offering an alternative explanation as to why they couldn't clone adults and why the efficiency dropped off as nuclei were taken from later stages of development. And they suggested, in effect, that the mechanisms which control gene function in order to produce different tissues were so complex and so rigidly fixed that they couldn't be reversed. And I think this is still a view, despite the fact that we now know it's not true, this is still a view that influences our thinking an awful lot, particularly older members of the community like myself. Because we were used to this idea that things were irreversibly determined, and it was not possible to reverse them. And maybe later on in the talk there'll be times when I might encourage us to be thinking, well, actually, is that true? Can't we push boundaries back even more than we have done already? Can't we make the whole process of changing cells from one type to another more efficient? It was at that time, roughly, same era, when this diagram, which I imagine many of you have seen, was put forward by Conrad Waddington, who was a research worker in the University of Edinburgh. It's epigenetic landscape. It's one of his writings about the epigenetic mechanisms which regulate gene function and so regulate development. What he put forward was the idea that this is a cell here whose fate was determined by, as it were, the outside mechanisms, the sort of regulatory mechanisms which said